Hey everyone, welcome back to my channel. So a lot of you guys, more than I can imagine, requested a video for basic immunology, especially that this is a very high yield topic. I saw a lot of immunology questions on my test. So hopefully by the end of this video, things get a little clearer. I'm gonna give you as much as I can of the basic immunology to try to outline this in your head. So let's get started. Alright, so the first thing I want to talk to you about, guys, is innate immunity or natural immunity that is present like ever since you're born. So these include barriers and cells. All of what you see in front of you here are things that prevent infection on a daily basis. So without these barriers, you would have been, infected every day. So these are stuff that prevent microorganisms from, getting into your body to start with. And another part of innate immunity are cells, which eliminate microbes once they get into your body. And again, those will also stop infection. So I want you guys to imagine with me this microorganism. And this will show you the difference, guys, between adaptive immunity and innate immunity. So when this microorganism gets in, your body will recognize it as non-self. So it differentiates it from your self-antigens. This will be innate immunity. It can only tell whether this is self or non-self. You can see here how non-specific it is, because it's rough. Adaptive immunity, on the other hand, as the name suggests, it adapts to a specific antigen, which means it can identify whether this bacteria, for example, is E. coli or is it staph or is it strep. You can see here how specific it is, and it will start making an immune response according to the specific antigen. So let's say, for example, all of these are non-self. The violet ones, all of them are non-self. That's what innate immunity can say. But adaptive immunity will make antibodies specific to these yellow ones, the epitopes. So that is the difference between innate and adaptive immunity. You can see here why adaptive immunity takes time and needs bridging by innate immunity. So the innate immunity is the immediate first line of defense, but we need time for adaptive immunity to develop, which is obviously more robust because it's more specific. So let's imagine together that here is an incoming bacteria and here is an epithelial barrier, which is considered one of the barriers of innate immunity. Even if this organism crosses this barrier and enters the body, will still find cells of the innate immunity, like phagocytes, right? So innate immunity is barriers that prevent infection and cells that eliminate microbes. And these cells will start secreting cytokines, which have a systemic effect, for example, fever, etc., on the hypothalamus, and they have a local effect, which is acute inflammation, the hallmark of innate immunity. They will bring in phagocytes and will bring in more immune cells to combat the infection on site. As you can see here, it is fast, it's immediate, first line, but at the same time it's non-specific, as I showed you, and I'm going to show you now how it's non-specific. And number 3, most importantly, and this is something that differentiates it from adaptive immunity, is that it has no memory. So no matter how many times the same organism enters your body, it will get the same reaction, acute inflammation. It's like innate immunity has Alzheimer's. It doesn't remember that it has encountered this antigen before. Unlike adaptive immunity, where you have the basis of vaccines, that once you're exposed to the antigen for the first time, you get a primary response and then you start getting symptoms. But once you're exposed for the second time, your immune system already remembers, already has memory. This part of the immune system is the adaptive one, and the secondary response is much more robust, and you wouldn't even feel that you got an infection or the organism entered your body again, because now you're ready. And that's not the same at all with innate immunity. So a key to the non-specificity of innate immunity will be the PRRs, pattern recognition receptors, like the toll-like receptor, that recognize, universal antigens on non-self antigens, called PAMPs, pathogen-associated molecular patterns. So, for example, all gram-negative organisms have lipopolysaccharide. So, this lipopolysaccharide is considered a PAMP, pathogen-associated molecular pattern, that is the same in all these types regardless of the species, right? So, your body, the toll-like receptor, which is non-specific, will recognize, oh, this is one of the PAMPs, so this is non-self. That's how non-specific it is. So this picture depicts the difference between, innate immunity and adaptive immunity. For innate immunity, as you can see, all of them share, like all of the classes of microbes, regardless of species, as long as they're all gram-negative, they share LPS, lipopolysaccharide, the orange one. So this is what's recognized by the PRR, the toll-like receptor. These are what's recognized by the innate immune system. But the adaptive immune system recognizes specific epitopes. It can differentiate E. coli from Klebsiella, from C. diff, etc., through these colors. So you have a distinct antibody for the green one, and a distinct antibody for the blue one and for the violet one. And because it's specific, it needs time. So until the adaptive immunity develops, the immune response, the innate immune response, will try to control the infection. Obviously, it is less robust, so sometimes highly virulent organisms still need adaptive immunity, but sometimes innate immunity can do and just finish off the infection. So let's talk about adaptive immunity. Once I say adaptive immunity, remember lymphocytes, all right? Because innate immunity, you have phagocytes, you have mast cells, basophils, this stuff. But when I say adaptive immunity, remember lymphocytes, whether B or T lymphocytes, okay? So this slide will highlight to you the difference between humoral and cell-mediated immunity. If we have an extracellular pathogen, for example, a bacteria that lives outside the cell, we will need antibodies in serum to, attack it, right? This is called humoral immunity. Humerus for serum. It's Latin for serum. So here we need antibodies in serum produced by B lymphocytes to attack it. But what if this pathogen, maybe a virus for example, is intracellular? It lives inside cells. In such a case, in order to kill the virus, I have to kill the cell that it has infected. So this is called cell-mediated immunity. And this is mediated by cytotoxic T lymphocytes. So cell-mediated immunity is for intracellular pathogens. Humoral immunity is for extracellular pathogens. And I mentioned this before in my immunodeficiency video, in how you can differentiate different primary immunodeficiencies based on whether it's a problem with adaptive or cell-mediated or humoral immunity. Now, both types of adaptive immunity, whether humoral or cell-mediated, they need T cell help. They need the help of T helper cells. The T helper cell subtype 1 helps cell-mediated immunity. It helps the cytotoxic T cell. The T helper subtype 2 helps the B lymphocyte. And we're going to see this now. Okay, so this is an overview of adaptive immunity. So let's get into it. So I want you guys to listen to this piece of info that T cells only recognize protein antigens. So we'll not be able to recognize any antigen that is not a protein. And there's a high yield caveat here in that they add proteins to vaccines Like if the vaccine antigen is polysaccharide, for example, they attach a protein antigen to it so that it can become more antigenic, that is recognized by T lymphocytes, right? Because T cells cannot recognize anything but protein antigens, while B lymphocytes can recognize all types of antigens, polysaccharide, protein, whatever, and they have antibodies on their surface that will be, once they are found to be specific to a certain antigen, they will activate the cell. So it's simple, right? So here is how a B cell exists in serum, by the way. A naive B cell that has never encountered an antigen before has IgM and IgD on its surface. These are surface antibodies. So once they recognize their specific antigen, which could be a polysaccharide one, for example, right away they start differentiating to plasma cells and produce antibodies. So you might think, why do we need T cells, right? I told you we need T cell help for any type of adaptive immunity. You can see here that the B cell, it recognized the polysaccharide antigen and did not depend on a T cell, simply because T cells can only recognize protein antigens. But you can see here that it can only produce one type of antibody in this method, which is the IgM. And this is characteristic of the primary immune response. Here you have no memory whatsoever. You're just going to have the IgM. It's going to stay for some time, and then it's going to go away, and we do not have any memory. Plus, it's not really high affinity, and it's only one type. But what if we have a protein antigen? In such a case, the CD4 T cell will be able to help the B lymphocyte. And what do we mean here by help? We mean here that we have different classes of antibodies. It's called isotype switching. Here, instead of doing just IgM, no, we have IgG, IgA, IgE. We have different types of antibodies, and the IgG, for example, is long-lived, right? In addition, the helper T cell helps make memory cells, B memory cells, so that when we encounter the antigen a second time, we're able to combat it, right? Also, by the way, the B cell needs T cell help with protein antigens, because with a protein antigen, as you can see, it's folded, so the antigens are hidden inside, unlike the polysaccharide that's stretched like this. So it actually needs the T cell for this reason. So as you can see here, we have different B cell clones depending on the antigen. So if we encounter antigen, the blue antigen, we're going to have a lot of those blue clones. If we encounter a green antigen, we're going to have a lot of green clones. So the cells increase in number in response to the specific antigen, so that when we have a secondary immune response, it's actually bigger than the primary response. It's stronger because we have more cells of this clone specific to this antigen. And this video shows it very nicely. You can see here that this is a specific type of antigen, and I only had one B cell. Once it recognized a specific antigen, we started having a lot of B cells of the same clone that is specific to this antigen. Imagine if this antigen enters a second time, it's going to have all those cells trying to attack it instead of just one cell. And that's how adaptive immunity has memory and is better than innate immunity. If this were innate immunity, it would react the same way, as if it has never seen this antigen before. Every time it reacts at the same intensity. But the adaptive immunity, every time it reacts in a higher intensity. And this is how the CD4 T cell helps the B cell to differentiate into plasma cells, isotype switching of antibodies, and make memory B cells, and that's most important. Alright, I told you guys before that T cells require a protein antigen in order to work. So we need the antigen to be protein in nature in order for the T cell to recognize it. But the T cell receptor also requires that this protein antigen be attached to MHC, major histocompatibility complex, in order to recognize it. So the T cell requires a protein antigen in association with MHC. Now, if this T cell is CD4 positive, it needs MHC class 2. And MHC class 2 is only found on antigen-presenting cells like B cells, dendritic cells, and macrophages. Now, antigen-presenting cells like these are the ones that phagocytose the antigen, they engulf the antigen, they process it, and then present it on their surface, like this, which means that they engulf the antigen or the organism in vesicles and then process these antigens to make it more, to break it down or assimilate it in order to become into small pieces in order to be recognized by the T cell. And so the antigen here is actually inside vesicles and then will be associated with MHC class 2 in order to be recognized by the T cell. And we've already agreed that this is protein in nature. Now, when this antigen presenting cell is a B cell, then it's the T helper 2 that will help it. If this antigen presenting cell is a macrophage, then it's the T helper 1 that will help it. T helper 2 helps B cells to class switch antibodies, while T helper 1 helps macrophages in order to move into cell-mediated immunity. So we can say that T helper 2 helps humoral immunity and T helper 1 helps cell-mediated immunity. Now, I told you guys before that MHC class 2 is only found in antigen-presenting cells, which engulf the antigen in vesicles, while MHC class 1 is found in all nucleated cells. And this is the one that will be recognized by the cytotoxic T lymphocyte. So if we have an infected human cell that has a virus inside, this will have MHC class 1 presenting the antigen inside it, and that's what will be recognized by the CD8 positive T cell. So if we have an antigen inside a vesicle, which would be the case in antigen presenting cells, it will be presented on MHC class 2 and recognized by CD4 T cells. If this antigen is kept into the cytoplasm, any nucleated cell, including antigen-presenting cells, will also be able to present it on MHC class 1. And in such a case, it will be recognized by CD8 T cells. So here is an example, guys. I have an antigen-presenting cell that has an organism inside vesicles. This will be recognized by CD4 T cells, because if the organism is in vesicles, it will be presented on MHC2. And the T cell type 1, T helper 1, will help the macrophage kill the microbe. While if this microbe is in the cytoplasm, which would be the case in most cells, then it will be presented on MHC1 and recognized by CD8 T cells and then killed. But remember that CD8 T cells also need CD4 help. So let's move on to cell-mediated immunity. Like I said, if you have a cytosolic antigen, maybe a virus, for example, that infected a hepatocyte, this is foreign, and so it will be presented on MHC1 to the CD8 T cell and will kill it. Now, you might think, what's the role of the CD4 T cell if the CD8 can just kill it? Now, without the CD4 T cell, the CD8 will not be able to differentiate and kill the infected cell. So without T cell help, CD8 can do nothing. Without T cell help, the CD8 cannot become memory cells, which means if you get exposed to this virus again, it will never know. For example, people who got hepatitis A for like once, if they're not vaccinated, if they get it a second time, they will not feel anything. Why? Because we have memory. Who let the CD8 T cells, who induced differentiation of CD8 T cells into memory cells so that they can recognize a second infection? It's the CD4 T cells. So here again, we have phagocytosed microbes in vesicles, and we have those in cytosols. Those in vesicles will be presented in MHC2 and recognized by CD4 T cells, which will produce cytokines like interferon gamma that will help this cell, which is a macrophage, kill the virus. Now, what if we have another microbe after killing the ones in phagolysosomes? We still have ones in the cytosol. Those will be presented on MHC1 and recognized by CD8 cytotoxic T lymphocytes. And with the help of CD4 as well, they will be able to kill the infected cell. Here's this picture, shows you how the CD8 T cell would be with and without a CD4 T cell. With a CD4 T cell, the entire process takes place and the CD8 becomes functional. But without the CD4 T cell, as you can see, there is no second signal for the CD8 T cell. I'm going to talk to you about this now. And so the CD8 T cell is exhausted and is unable to have antiviral activity. So any T cell, guys, requires two signals to become active. It's not enough that the T cell receptor recognizes a protein antigen in association with MHC. This is just one signal. This is called signal 1. Signal 2 is to have a co-stimulator in association with the receptor. If both are found, signal 1 and signal 2, the T cell will be active, whether it's CD4 or CD8. If we have only one signal, which is MHC in association with the protein with the T cell receptor, without co-stimulation, then the T cell becomes anergic, as in it's exhausted and wouldn't work. And that is the case with CD8 T lymphocytes when there is no T cell help. Now, what exactly is this co-stimulatory signal? Signal 1, like I said, is the MHC class 2 in association with a protein antigen. Signal 2 for any antigen presenting cell except the B cell will be CD28 receptor on the T cell and B7, this by the way equals B7 on the antigen presenting cell, which could be a dendritic cell or a macrophage. Now, what if it's a B cell? If it's a B cell, the co-stimulator will be CD40 on the B cell in association with CD40 ligand on the T cell. So any other cell, for any other cell, the co-stimulator signal is CD28 on the T cell, B7 on the other cell. Except the B cell, we have CD40 on the B cell, CD40 ligand on the T cell, and you get asked on this, by the way. Alright, so here again, with those two signals, signal 1 and signal 2, the T cell will be able to work. And so if this is a tumor cell that we want to kill, and this is, for example, a CD8 T lymphocyte, signal 1 is there, and with the help of CD4 T cells, we have signal 2 as well. And so in such a case, the T cell will be active and will be able to kill the tumor cell. Now, it's actually important to have this requirement of two signals instead of just one to prevent autoimmune disease. Because imagine we have resting antigen presenting cells in the blood floating like this, and they engage with a naive T cell. If it were just one signal requirement, you will get autoimmune diseases and you will get excessive unnecessary T cell response. But only when the antigen presenting cell is activated by microbes, you will get a co-stimulatory molecule will come out on its surface and then will provide the second signal for T cell activation. In such a case, it will be a true signal, right? And a high yield caveat here that has just been discovered recently is instead of this second signal, tumor cells actually started bringing out something that is an inhibitory signal. So when we have an inhibitory signal like the PDL1 instead of the signal 2, that means we will have energy because it's as if I only have signal 1. And for this reason, we have immunotherapy, antibodies that attack this interaction so that we can allow signal 1 and signal 2 to act together. So here is an example, guys, of signal 1 and signal 2 together activate antigen-specific T cells. Without signal 2, the T cell becomes anergic. Without signal 1, if you only have signal 2 and no signal 1, also there is energy. Finally, guys, I want to mention the natural killer cell. The natural killer cell is very similar to the CD8 T cell, but it's part of innate immunity, as in it's non-specific. Now, normally, under normal conditions, the natural killer cell does not attack a cell as long as MHC class 1 is expressed. So we have an activation receptor and we have an inhibitory receptor which inhibits the NK cell from attacking as long as MHC class 1 is still there. Once MHC class 1 disappears or is down-regulated, then the activation signal is, now active, nothing is inhibiting it, and then the NK cell will try to kill this cell. Do you know why? Because one of the virulence factors of viruses is that they try to down-regulate MHC class 1 to avoid recognition by the cytotoxic T lymphocytes. But not so fast, because the NK cell is there and recognizes this trick, and in such a case will be able to kill this cell because it knows that this cell must have been infected with a virus in there that's down-regulating the MHC. Like, how cool is that? I hope this video helped, guys. I gave you a general outline of the high-yield basic immunology you might be asked about. Let me know what you think in the comments. All the best, guys.